Hello everyone, in this final lecture on biophysics, we'll talk about acids, bases, and buffers. So life is basically a chemical system. It is a complicated series of molecules intertwined and organized together as directed by the information in DNA. But in order for this system to function optimally, we need to maintain the internal environment in a process known as homeostasis. And these are the parameters that are regulated in homeostasis, such as temperature, osmotic pressure, pH, which is our topic of discussion today. Okay, these are the learning outcomes for this session. Basically, we'll discuss everything on pH and acidity. Then we'll look at how we can use this parameter pKa, or the acid dissociation constant, to predict the strength of an acid. And you will learn why weak acids can serve as buffers. And last but not least, you will learn how to calculate the pH of a buffer by using this equation. So, why should we care about pH? Why is it so crucial to life? Well, we know that pH measures the level of acidity and basicity of a solution. Because we are so sensitive to minor changes in pH, even a deviation of more than 0.5 can kill us. So showing here, you have the normal range of pH. We call it the physiological pH, which is in between 7.35 up to 7.45. In other words, it is 7.40 plus minus 0.05. Any deviation from that value will result in either an acidosis or alkalosis, and any bigger deviation, it will kill us directly. So before we proceed, here is an important concept that you need to understand. We know that water are highly polar molecules, which means they can form partial charges, such as delta positive on the hydrogen atom and delta negative on the oxygen atom. But a tiny amount of water molecule can actually react to one another, forming these charged ions, such as the positive hydroxonium ions and the negative hydroxide ions. So as these ions are charged, they are highly reactive. Just for information, in most textbooks that you read, the hydroxonium ion is commonly abbreviated as H positive, or the hydrogen ion. But the fact is, H positive ion does not exist in water. It is just a convenient shorthand. So please bear in mind, whenever you see H positive ion, it should be H3O positive, or the hydroxonium ions. Because the dissociation of water molecules is a reversible reaction, it means we can actually calculate the KEQ, or the equilibrium constant of this reaction, which has the following equation, which is H positive times OH negative divided by H2O squared. Again, the H positive here refers to H3O positive. Now, to know the values of H positive and OH negative, we have to perform an experiment to find them out. But to know the value of H2O is rather easy because it can be derived on paper. So what we need to do is to find out the concentration of H2O, which is its molar concentration. We know that 1 liter of water weighs just exactly 1000 gram, and it has a molar mass of 18 gram per mole, which means 1000 divided by 18, it will give us 55.6 moles of water in 1 liter, and that is equal to a molarity of 55.6 molar. But in practical, instead of using KEQ, scientists are using KW, or the water equilibrium constant. That's because the water molecule is vastly in excess in almost all aqueous solutions, and therefore we can safely assume that the H2O is a constant. So by removing this part from the equation, it gives us Kw is equal to H plus times OH negative. Again, the H positive here refers to H3O positive. So Kw is a constant under specific temperature. From experimental observation at 25 degrees Celsius, Kw is equal to 1 times 10 to the power of negative 14 molar squared. In other words, the product of H3O positive times OH negative at 25 degrees Celsius is equal to this value. Since we have equal amount of H positive and OH negative in pure water, as you can see from this reaction, then H positive is equal to 10 to the power of -7 molar. So you can see that in pure water, the concentration of free hydrogen or hydroxide ions is quite low because we are talking about a value with 6 zeros after the decimal point. So acids and bases are the chemicals that can change the concentration of H positive in a solution. An acid is defined as a substance which produces hydrogen ions by dissociation, such as HCl, while a base is a chemical that releases hydroxide ions, such as potassium hydroxide in this case. So adding acids or bases is a fundamental way to change the amount of H positive available in a solution. The amount of H positive in acid or base can be very different. It is about 0.1 molar for strong acids, and it is 10 to the power of -14 for strong bases. So in order to convert these values to something more convenient and more making sense to us, we employ the pH scale, which is based on the negative log of H positive concentration. So why is it logarithm or exponent? Because it can manifest big values with very small numbers. And here is just a quick revision of your basic mathematics. For example, 10 times 10 will give you 100, which is 10 to the power of 2 or 10 squared, while 1000 is equal to 10 times 10 times 10, which is 10 cubed. So instead of big numbers here, by using the exponent, we are getting 2 and 3, which are the power in small number. On the other hand, if you are multiplying these figures together, you can actually add the power if the base is equal. In fact, exponentials are very common in biology. So let's just take the example of bacterial growth. From the very first parental cell, upon cell division, you'll get 2 daughter cells. And then from 2, it gives you 4 cells, and so on. So the general formula of number of cells is equal to 2 to the power of t, where t is the number of division cycles. If you were to plot a curve from this general formula, what would be the shape of this curve? Well, it is known as an exponential curve. So this happens to cells, it happens to prions, invasive species like viruses, as long as you have a constant replication rate. We have seen exponents in whole numbers, but actually exponents can also be less than 1, such as an exponent of 0.5 is actually a square root, while an exponent of 0.333 is a cube root. Not only this, exponents can also have negative values. For example, 1 over 10 is 10 to the power of -1, or 1 over 100 is 10 to the power of -2. And again, you can add exponents when you're doing multiplication, or you can subtract the exponents when you're doing division, provided that the base is identical. And next, we'll look at logarithm. Logarithm is actually another way of writing exponents, and it is known as the inverse of exponents. So over here, you have a log of base 10 with a value of 0.1, so it has a power of -1. And here, log 10 with a value of 0.01, it has a power of -2. So on and so forth. And you can easily use your calculator to tell the answer. For instance, log 10 5000 is equal to 3.69, which means 10 to the power of 3.69 will give you this value of 5000. With the use of log 10 scale, we have a practical scale of pH because the log 10 scale has just compressed everything. So instead of having many zeros, which is difficult to make sense, we use smaller numbers to represent the same value. So what are other logarithmic scales that you can think of? You have something such as the Richter scale in geology for measuring earthquake. So that is a log scale as well. Okay, here are some basic rules about logarithm. When we say log 10 of something is equal to a, then a is the power of that value with a base of 10. So you can look at this example yourself. And when we have the multiplication of two values within a log, we can actually break it into two individual logs. So log 10ab is equal to log 10a plus log 10b. Here is an example. So division is opposite. Log 10a divided by b can be broken down to log 10a minus log 10b. So you can just refer to this example yourself. And here is a practice question. Please pause the video for a while. And the answer is D. Please consult your friends or go back to your mathematical textbook if you have any questions. So far, we have talked about log with a base of 10, but in fact, you can have log with any base, such as a log with base 5. But in science, the most common log that you will encounter is the natural log with a base of e. So what is e? It is a mathematical constant known as the Euler's constant, and it is an irrational number with a value of 2.718, and it is never ending. So it pops up a lot of mathematics, such as pi, which is also another constant that we use. So the natural log is written as log ex, and it is also known as ln x. So you should find it familiar because it is used in the equation of Gibbs free energy. Over here, can you see ln? Because you have a base of E, which is the Euler's constant. And how could you know the values of E? Well, you can find a dedicated button on your scientific calculator. Now let's come back to our pH scale. Remember, pH is defined as negative log H positive. And as I explained earlier, that could be a wide range from 10 to the power of negative 1 up to or down to 10 to the power of negative 14. So to compress this scale, we use log with a base of 10. And that gives us a pH scale from pH 1 to pH 14, 1 being strong acid, 7 neutral, while 14 being strong alkaline. Again, what is our physiological pH? Yes, it is at 7.4, which is slightly alkaline. So why is pH important? Well, the answer is that it is linked to enzyme function. We learned that enzyme function is dependent on its structure, and enzyme structure is maintained by different types of intra and intermolecular forces such as hydrogen bonds, ionic interactions, and so on. So what pH does is that it can change the ionization pattern or the charges on the surface of the enzyme functional groups, such as the carboxyl group in this case. And therefore, with a different pH, we might change either the structure or the surface charges on the enzyme, and that can render the enzymes useless. In the following section, we will discuss about weak acids, which dissociate partially to form the hydroxonium ion and the conjugate base. Because this process is a partial dissociation, it means it is reversible. And for any reversible reaction, we can always calculate its equilibrium constant. And in this case, it is known as the acid dissociation constant, Ka, which has this formula of conjugate base times H positive divided by the weak acid. Here is a very quick example. So under standard conditions, which is 1 atmospheric pressure and a temperature of 298 Kelvin, the Ka for ethanoic acid is 1.8 times 10 to the power of negative 5 molar, which is a very small number. So how do we make sense of it? Again, we use logarithm to compress the decimal scale, and we end up using pKa, which is a parameter to tell us how strong an acid is. So here is the formula. pKa is equal to negative log 10 Ka. So if a solution is having a pKa of less than 3, that is a strong acid. Anything in between 3 and 7, it is a weak acid. 7 to 11, a weak base. More than 11, it is a strong base. So whenever pKa is small, it means it is an acidic solution. Why is it so? Because you have a negative sign here, which means when this value is small, Ka must be big. And if Ka is a big value, it means you have more conjugate base along with the hydroxonium ions. So the more H positive you have, it means it is a more acidic solution. Okay, these are just examples of acids and bases. HCl, for example, has a pKa of -7, so it is an extremely strong acid. On the other hand, the ethanoic acid is around 4.7, a weak acid. Ammonium or ammonia has a pKa of 9.2, so it is a weak base, while NaOH has a pKa of 14, which is a strong base. And we can actually apply the concept of pKa on amino acids because basically they are weak acids. That is why we call them acids. So if the pH of the environment is lower than pKa, meaning that the environment has more H positive, what will happen? Well, the reaction will shift towards left hand side so that you will get more of the weak acid production. Conversely, when the pH is higher than pKa, the reaction will shift towards right hand side so that it will get more conjugate base formation. And when pH is equal to pKa, half of the molecule will be weak acid while another half will be in the form of conjugate base. So why is this information important? Because it will tell us what will be the charges on the molecule in vivo. It is important because we can make prediction based on the pKa and pH of the environment. Okay, showing in this diagram, you have the basic structure of an amino acid, and you have four different groups, including hydrogen, that are attached to the central alpha carbon. For both the amino and carboxyl groups, they are capable of protonation and deprotonation. In other words, they can either gain or lose its proton. And depending on the type of the functional group on the side chain, some R group are able to protonate and deprotonate as well. Now here is the important thing. For every functional group that is capable of protonation and deprotonation, we will assign them with individual pKa values. And you will understand what I mean by answering this question. So please pause the video for a moment. And the answer is C. You have three groups or three pKa values. So what are those three groups? The first one, carboxyl group, and then the amino group and another carboxyl groups. They are all capable of protonation and deprotonation, and therefore we assign each of them with an individual pKa value. Okay, here is an amino acid with three pKa values, which is aspartic acid. So these are the three pKa values at 2.1, 3.9, and 9.8. So when the pH is lower than 2.1, which means the environment is more acidic, all those excessive protons will be uptaken by both functional groups as well as the R group. It means all functional groups are fully protonated. When pH is between 2.1 and 3.9, so one functional group will deprotonate. In this case, it is a carboxyl group. And when this happens, what you get here is a pseudo ion, as there is no net charge on this molecule. When the pH is between 3.9 and 9.8, another carboxyl group will be deprotonated, so it becomes a mononegative molecule. While when the pH is higher than 9.8, which means the surrounding is very basic, all of the functional groups will be deprotonated. And in this case, this amino acid has become a dinegative amino acid. As a matter of fact, all amino acids have at least two pKa values, and depending on the side chain, you might have the third pKa value. Okay, here is a question to test your understanding. Please pause the video for a moment. And the answer is E, 11. Why is it so? It's very simple. When will glycine become a negative ion? When it loses its proton. And when will it lose its proton? It's only when the environment is more basic than the pKa2, which is any value that is higher than 9.6, which in this case is E. So what you have witnessed from the previous example is that amino acid can function as a buffer, which stabilizes the pH of a solution. So living organisms, we depend on buffer to prevent large pH fluctuations within our cells. So what are buffers? They are basically chemical mixture which consists of conjugate acid and conjugate base. For example, in our blood, we use the carbonic acid slash hydrogen carbonate system. So here in this reaction, which one do you think is the conjugate acid? Well, it is the carbonic acid. And which one is the conjugate base? It is the hydrogen carbonate ion, or better known as the bicarbonate ions. And in this system, you have a pKa of 6.1. When the carbon dioxide produced by our cells in metabolism dissolves in the blood plasma, it becomes carbonic acid with the help from the enzyme carbonic anhydrase. And we all know that carbonic acid is a weak acid, which means it dissociates partially into H positive and HCO negative with a pKa of 6.1. Again, the H positive here actually refers to H3O positive. So if you add strong acid into this system, the extra H positive will be absorbed by the conjugate base, forming up more carbonic acid. Conversely, if we add a strong base to counter the change in pH, the carbonic acid will dissociate to release more protons so that they can combine with OH negative to form water molecules. So within a specific range of acid and bases added to this system, you are going to get an almost fixed value of pH. And this region of fixed pH is known as the buffered region. A buffer could work as long as you have a reversible reaction producing weak acid and the conjugate base. And it works simply according to the Le Chatelier's principle. Okay, showing here is a visual explanation of pH buffering. So to become a buffer, it must be a weak acid that dissociates partially, which means you are always getting this reversible reaction. So what will happen if you add a strong base into the system? Well, adding base means you are adding extra OH negative to the system, and to counter that change, this OH negative will be absorbed by the weak acid, forming up water molecules. Now, speaking in more details, the HA or the weak acid will dissociate to form the conjugate base, while the H positive will combine with OH negative to form water molecules. So from this picture, you can see that you definitely have more water molecules after the reaction. And as a result, pH changes only very slightly. Conversely, if you are adding a strong acid, you are adding in more protons into the system, and those protons will be absorbed by the conjugate base, forming out more weak acid HA. So from this picture, you could see that there are more weak acid HA after the reaction. Again, pH changes only very slightly with the buffering effect from this reaction. Well, actually, we can prepare a buffer of any pH by referring to this equation, which is known as the Henderson-Hasselbalch equation, and it is derived from the acid dissociation constant Ka. So what we do is that we put log on both sides of this equation, so it becomes log Ka is equal to log H plus log A minus log Ha. Remember, multiplication is plus while division is minus. And you have already learned that pKa is negative log Ka, and therefore log Ka is negative pKa. Again, you know that pH is negative log H positive, and therefore log H positive is negative pH. So by rearranging the formula, you'll get this final equation, which is pH is equal to pKa plus log A minus divided by Ha. And we can use this formula to calculate the pH of any buffer. Here is an example of the carbonate system in our blood. It is given that the ratio of hydrogen carbonate versus carbonic acid is about 20. Now, what is the normal pH of our blood? Okay, here is the formula. So by plugging in all the information, it will give you pH is equal to 6.1, which is pKa of this system, plus log 20, because we know that the ratio is 20, and that will give you pH is equal to 7.4, which is our physiological pH under normal conditions. And here is the practice question. Please pause for a moment. And the answer is D, 40. So make sure that you know how to use the equation. You do not have to memorize the equation, but when the equation is given to you, you must know how to calculate correctly.